The bithorax and the trithorax. So basically, two types of protein complexes came out of this. Are there other people missing, right? They're coming in. Okay, we'll just get started. So basically, of those phenotypes that we're talking about, the homeobox gene phenotypes, when you started to see these multiple limbs, fins are essentially limbs, and or you start to see other elements come through because of the lack or activation or repression of Hox genes. You saw different phenotypes. You saw which genes suppressed them, which genes activated them. You came up with two sets of protein complexes, one called polycomb 1 and polycomb 2, so polycomb group of complexes. The key of that was enhancer of ZEST, EZH1, EZH2. That's a histone H3K27 trimethylation complex, methyltransferase. Okay, so that's how they found out that EZH2 is a major repressor of homeobox genes that does it through H3K27 trimethylation, and therefore an epigenetic mark was born to be H3K27 trimethylation. Okay, is that clear? So the previous one that I showed you in the eye color was suppressor of variegation. That was SUV39H1 that did H3K9 trimethylation. That was heterochromatin, long-term silencing. Then there was EZH1, EZH2, those genes that started to do H3K27 trimethylation. Those are two major repressive marks in histone terms in our body. H3K9 trimethylation, H3K27 trimethylation. Clear? The first one is done by SUV39H1, and there are other proteins called ZDB1, etc. And then these were done by EZH1, EZH2. Those are polycomb group of proteins. Polycomb group of proteins do K27 trimethylation. That is found in a lot of genes where those genes are silent, in many of the tissues where it should not be expressed. Remember the example of aldehyde dehydrogenase, a neurotransmitter. So that is silenced by K27 trimethylation, not by K9 trimethylation, okay, necessarily. Some genes are by K9, but K9 trimethylation is usually not very pervasive in silencing genes. It's mostly used for suppressing repeat elements, viral elements. Okay, is that clear? K27 trimethyl is more often used to silence genes that are genes, meaning for your specific purposes. K9 trimethyl is more often used for